Hi everybody, this video will be divided into two parts. The first part is an experiment in which we will be looking at a banana's DNA for you to better understand the topic. And in the second video, I will be explaining you the main parts of our two sections. They say people and bananas share 50% of the same DNA. So let's see a banana's DNA. You will need half a banana, alcohol, 1 Ziploc, 1/2 cup of water, contact solution, this is optional, 2 tablespoons of dish soap, and 1 tablespoon of salt. Before you begin, make sure you have chilled your alcohol in the freezer. This is a very important step because room temperature alcohol will not work nearly as well as cold alcohol. So, place your alcohol in the freezer for 30 minutes. Your first step is to put 1/3 of the banana into a Ziploc and start squishing it up until it turns into a paste like this. In a separate bowl, you will add 1/2 cup of water, 1 tablespoon of dish soap, contact solution if you want, and mix it up with 1 tablespoon of salt. Now add your solution to your Ziploc bag of squished up fruit. Add 1/4 of a cup of the solution. Flatten out your baggie to remove most of the air and then seal it up. Gently squish the liquid around. Let it chill for 20 to 30 minutes to give the detergent time to release a lot of DNA. Now with a coffee filter, we will pour the food mixture into a filter and add them into a cup. Now we will add the alcohol. You will want to use at least 70% or up alcohol for the extraction. The higher the concentration of the alcohol, the better it will work to make the DNA come out of the water solution. The DNA will come out of the solution at a boundary layer between the water and the alcohol. You now should see some white string, almost cotton-like strands, begin to appear in the glass. That is the DNA. You can collect the DNA using a toothpick, chopstick, or paper clip. First of all, we will be talking about Frederick Griffin, who was a British bacteriologist who was investigating 2 forms of the bacterium that causes pneumonia. The first form was the S form, which has a coating made of carbohydrates, and the second form was the R form, which didn't have a smooth coating. Now, let's talk about his experiment. He did a mice experiment, like you can see in the image, in which he expected only the S type to kill the mice. But when he injected both the S type and the R type, the mice also died. So he concluded that some material must have been transferred from the heat-killed S bacteria to the live R bacteria. So he called this mystery material the transforming principle. Now let's talk about 2 American biologists, Alfred Hershey and Martha Chase, who were studying viruses that infect bacteria. This type of virus was called a bacteriophage, like you can see in the image. This takes over a bacterium genetic machinery and directs it to more viruses. Hershey and Chase wanted to make an experiment to see of what was genetic material done, like you can see in the image. DNA or protein, so they made 2 experiments, testing proteins and testing DNAs. After they made these experiments, their findings finally convinced scientists that the genetic material is DNA and not protein. Now, the small units that make up DNA are called nucleotides. They have three parts that you can see in the image: a phosphate group, a deoxyribose, and a nitrogen-containing base. There are four types of nucleotides: the cytosine, the thymine, the guanine, and the adenine, and they differ only in their nitrogen-containing bases. Lastly, but not least, Watson and Crick wanted to know the structure of DNA. After the several models they made, in 1953 they finally published their DNA model, which was a double helix model. This double helix model, in which you can see in the image, in which two strands of DNA went around each other like a twisted ladder. The bases of the two DNA strands always pair up in the same way. This is summarized in the base pairing rules, in which thymine always pair up with adenine and cytosine always pair up with guanine.